Says is that if you add a solute to a solvent, the amount of pressure required to stop osmosis from occurring is going to be higher. And then the last one is another type of pressure. Yeah, actually, this one is sort of like a special case of number 2. This is vapor pressure lowered. So what this means is that if you can reduce or lower the vapor pressure of a liquid by adding a solute to that solvent. Okay, so let's go to question. We did questions 1, 2, and 3 last week. So question 4 states, you are lost at sea and have no pure water. Is it a good idea to drink seawater? Give reasons. Is this what those contestants do in Survivor? I've only watched, I think, like 5 minutes of Survivor in many years it's been on TV. Is that what they do in Survivor? If you survive, they drink seawater. They're so thirsty. They get thrown over. Is that how they get there? They get thrown over off some ship and have to swim to some island. Then they pretend they're on Gilligan's Island and. Oh, Gilligan's, you guys are too young to remember Gilligan's Island.
Good time.
Okay, so for question 4, what colligative property is involved here?
Osmotic pressure?
Yeah, so it's osmotic pressure, so it's a good idea to drink seawater. Okay, so let's see, what's happening there is if we draw a picture. Okay, so here's a cell in your body. And in your cell, there's salt, and the concentration of salt in your cells in your body is about 0.9%. Now the concentration of salt in seawater varies from about 3 to 7%. We'll say it's like 5%. Note that the concentration inside the cell and outside the cell are different. So since your cell membrane is a semi-permeable membrane, it's going to allow water to pass through into the cell or out of the cell. So in this case here, is water going to pass into the cell or will it pass out of the cell? Out. Okay, so you're saying that water is going to pass out of the cell, and if water passes out of your cell, what will happen to you if you drink seawater? You'll get dehydrated. Yeah, that's not good. So now again, another way you can look at this is in osmosis. Okay, so water passes through the semi-permeable membrane. So salt, okay, so again, in osmosis, water only passes through the membrane, not salt or sugar or anything else. Okay. Now, if you have a different type of membrane, then another application of osmosis, you have, for example, a dialysis membrane. In dialysis, water as well as small molecules will pass through the dialysis membrane. Okay, so you can have you can have water passing through and salt. And what's another example of a small molecule which can pass through here? You have potassium ions. You can also have calcium ions. Will glucose pass through here? Glucose pass through the dialysis membrane? For those of you taking biology? No? So you're saying, so dialysis, so you're saying that molecules, so large molecules don't pass through. So these large molecules, now they include proteins because proteins are very big. Glucose is a simple carbohydrate. So is glucose an example of a large molecule that doesn't pass through the dialysis membrane? Yeah, usually glucose will pass through. Okay, so glucose. But if you have something like starch, which is a complex protein, that's not going to pass through the dialysis. Okay, so getting back to this, in osmosis, note that if you have 0.9% NaCl, that means you would have, let's say, 99.1% water. And if you have a 5% salt solution, that means you have 95% water. So note that it's going from a high water concentration to a low water concentration. And that is the driving force for osmosis to occur, this difference in concentration. Okay, so this concentration difference is the driving force for osmosis. And the fancy word for difference is the concentration gradient.
The concentration doesn't change from like 99.1.
Yeah, so you're right. So as osmosis occurs, then the concentration inside the cell is going to change.
And does it become like, does it return to its previous state after a while or something? Like the sodium when you take salt from it?
No, so what happens? Yeah, so what happens is that the osmosis will occur until these concentrations of these two solutions are the same. Because note that as water passes out of the cell, is this concentration going to increase or decrease? Okay, so remember from the concentration, one of the concentration in molarity is moles of solute per liter of solution. Note that as the volume or as the amount of water changes, note that what's happening here? Is it increasing or decreasing? It's decreasing, so note that as the denominator gets smaller, the concentration is going to increase. Okay, so as water passes out of the cell, this is going to increase from 0.9% to a higher concentration. What happens to the 5% NaCl solution? That's going to increase or decrease? That's going to decrease the concentration. Okay, so osmosis will stop when those two concentrations are the same. Okay, and if you're lost at sea and you're really thirsty, what should you do? Yeah, that's one thing you do. Although, see, one problem with drinking urine is that there's urea in your urine, and actually urea isn't really so good to drink. It's sort of toxic. That's probably your last resort. Yeah, so if you're sitting out in your little rowboat, your little life raft in the middle of the sea, and you're really thirsty, and that sun is beating down on top of your head, how are you going to get fresh water?
Water has a lower boiling point than salt water. So
remember you guys did this in Chem, your Chem1B Lecture? We took a water, this is your seawater. Boiled water. That's your seawater, so what you can do is you can put like some, maybe you have some saran wrap, here's a rock, here's some plastic wrap. Here's the sun. And then you just put a little cup in here. So then the sun is going to evaporate the water, and it's going to form these little beads of bubbles, and then it's going to condense into your fresh seawater. Just in case you're ever caught in some life raft.
So if you have plastic wrap, and like, you know,
see, the water condenses on the plastic wrap. They put a little rock in it, so it all should dribble down the middle.
Yeah.
Actually, see, I came up with this Chem1B lab when I actually saw this in some article. Okay, let's go into question 5. Salt is found in vegetables and in fruit. How can limp lettuce be made crisp again? So don't you hate it when you got your fresh, you picked out a fresh head of lettuce at the store and you bring it home and you want to make your salad and you make your salad and you take it out of the refrigerator and the lettuce is all limp. You know, you feel like, then what often people do is they start peeling off those leaves of lettuce till they get down to the crisp pieces, and the next thing you know, you've peeled off every piece of lettuce and you throw the whole head away. Ever happened to you?
Yeah.
So how do you make that lettuce crisp again?
Put it in water.
Have you guys done this lab in biology? An osmosis lab like this in biology? You use either lettuce or celery or I don't know, cucumbers or. You guys haven't done that?